Ladies and gentlemen, please welcome to our last session for today's conference. Our last speaker is Mr. Indra. He is a doctor of philosophy, candidate at Jenner Institute, University of Oxford. We should be very proud of him because he is one of the AstraZeneca team. Mr. Indra, are you already in here? Oh, yes. Good morning in UK.
Hello, good morning, good afternoon for Indonesia.
We can't hear your voice.
Hello. Can you hear me right now?
We still can't hear your voice.
Let me show.
Hello.
Hello, can you hear me right now? Hello.
Mr. Indra.
Hello. Hello, can you hear me right now?
Yeah, we can hear your voice.
All right. Hello.
Here.
Hello. Sorry, I can't hear the moderators.
Vaccine research and development covering antigen design, preclinical, clinical development, and manufacturing process. And he's currently studying in Jenner Institute, Nuffield Department of Clinical Medicine in University of Oxford. Mr. Indra has got so many work experiences, including internship, vaccine and technical policy, COVAX facility from March until August 2022, and then product developer, R&D, Biopharma Indonesia from 2014 until 2019. And he also involved in volunteering, like in clinical of Oxford malaria vaccine candidate. That is very interesting. And then vaccine advocacy campaign and consultancy. And he also vice president of Indonesia Students Associations in Oxford. Okay, Mr. Indra, can you start your presentation now?
Yeah.
Yeah. The screen is...
Hi, everyone. Good afternoon. I hope everyone can hear me. First of all, I would like to say thank you so much for the committee that has been kindly invited me to this event. I've been asked by the committee to talk about, to share about my experience during the clinical trials of AstraZeneca vaccines. One of the, I've been so lucky to get involved in the teams, and I learned a lot about how the vaccines can be translated from the preclinical to clinical trials and produced in a massive amount to be distributed to many people around the world. Of course, during that process, there is a lot of collaborations happens between one team to the others, between one organization to the others, and between countries to countries. Um, and in the previous sessions, most of the speakers has been elegantly talking about how the collaborations can be conducted in different fields, and of course, that's the collaboration has been a center of the work in each effort. And now I would like to talk about how the collaboration also become our heart in the vaccines research. So without further ado, let's move to the next slide. Um, as you know that we also the pandemics already happened for the last 2 years, but we still have vivid memories about how the virus SARS-CoV-2 emerged in the late of 2019 and escalate become pandemics in the middle of March. And probably most of you just realized that the virus can be very badly for us uh, by that times. But for us, for the scientists, we already know that this virus can be escalated into something bigger than just an outbreak, as because at the time we didn't have any vaccines or treatment for SARS-CoV-2, and based on the based on the clinical data from the China, we see that how badly people get infected by the SARS-CoV-2. That's why once the the genomic data of SARS-CoV-2 are available, all the vaccine developers start to develop COVID-19 vaccines. But we also not guarantee or confident enough to can make an effective vaccines, because based on the historical data, we know that vaccines development could take years to be developed. For example, like typhoid vaccines, it takes around 100 years since the pathogen emerged and until the vaccine is licensed. And of course, the other example is polio that already been very make us very devastated in the in the beginning of 19th century, and the vaccine just be available after 40 years of development. The record the record one is a vaccine for measles that only take 5 years to be developed since the virus emerged to the vaccine available in humans, but it takes 5 years to be developed. So we can't guarantee that by the time that the vaccine for COVID-19 can be available soon, but the pressure to to save humans' life and also to make an effective vaccine is quite big, as the pandemic is really make us devastated in many of sectors. Um, However, because of that pressure, the record of measles vaccine has been broken by the development of COVID-19 that only takes 1 year to be developed since the virus emerged to the vaccines available. Obviously, there is a significant situation where in the past the technology probably not as advanced as right now, and also the amount of funding that available for development of COVID-19 is massively big compared to development for any other disease that also could contribute to the acceleration of COVID-19 vaccine development. Yeah, to get on, but is it actually really possible to make a vaccines in the record times, since and also how to prevent the pandemic in the future? To be able to answer that, we need to know how actually the vaccines development process occurred. For us, first of all, since the pathogen is discovered, we need to design the vaccines based on the targeted of pathogen. It can be virus, bacteria, or parasite, depend on the disease, depend on the pathogen that causes the disease. Once we finish to design the vaccines, we then test the vaccine in the vaccines in animals, which call it preclinical study. we test is the vaccine immunogenic enough to induce immune response such as antibody or T cell response. If the vaccines can protect animal, if there is a sufficient model challenge to test the vaccines in animals. If not, then probably we need to directly move to human clinical trial to test if the vaccine effective or not. But in human itself, the clinical trial is divided into 3 phases: phase 1, phase 2, and phase 3. And the number of volunteers and the number of resources that require for each phase is escalated as well based on the increase of the number of phase of that we conducted. For example, in phase 1, the aim of the phase 1 of clinical trial is to test if the vaccine is safe and immunogenic. Usually it involves small number of people. Then if the vaccine has been proven safe, we move forward to phase 2 to test if the vaccines already is immunogenic in targeted populations. For example, in COVID-19 vaccines, the most vulnerable population is elderly and immunocompromised. So we involve those populations in the phase 2 trials. And if the vaccine has been proven immunogenic and safe in those populations, we also progressing to phase 3, which is to test if the vaccine has an efficacy to protect people from the disease, or if the vaccine can stop the transmission or not. So that's been evaluated in phase 3. Normally, since the beginning of the phase 1, all the clinical trial could take up until 10 years. Because we don't want to progress to the next phase if the previous phase is failed, because there will be economical loss, and also there is a risk that we need to consider when we move to the next phase. And also the clinical trial itself is not cheap, because it takes a lot of resource, it takes a lot of it requires a lot of volunteers. So we usually do clinical trials in a staggered process. So we finish phase 1 first, then we continue to phase 2 and phase 3. Once the vaccine has been proved as safe and efficacious, we then need to think about how to manufacture these vaccines, because if the vaccine works, obviously we need to manufacture it before it can be distributed to the main to the communities. So we develop the process how to manufacture the vaccines from lab scales into manufacturing scale, and it takes a lot of process as well, because we deal with the biological substance, which is not linear. When we're scaling up, for example, from 100 liter to 1,000 liter, all the parameters of the process need to be optimized against. Once we already manufacture, we need to license our vaccines, and we're working with regulatory, for example, with BPOM in Indonesia, and the BPOM will audit, will review our all document of manufacturings, and if the vaccine has proven sufficient, if the data is sufficient enough to prove that the vaccine works, and all the manufacturing process follow the highly regulated protocol that we call as GMP, good manufacturing practice, then the vaccines can be licensed and distributed to community. But that's the normal situation where the vaccines start from discovery to licensing. However, during the pandemic, obviously we couldn't follow the normal process. We need to accelerate the vaccine, and how is that possible? So after we got the data from preclinical trials, we moved to the clinical trials, and we do the clinical trials sequence in parallel. So for example, when we already established the safety in the phase 1, we don't finish the follow-up of the volunteer in the phase 1. We move to phase 2 to test the vaccine in the targeted populations. In parallel, then if the safety in the targeted population already been established, we also move to the phase 3, where we test the efficacy in the bigger community. So we need to de-risk about the financial loss if the vaccine doesn't work, as we need to make a vaccine as soon as possible. Also, in parallel, the team for process development start a study how the vaccines can be manufacturing in massive amount. So they study how the production can be scaling up, and also we working closely with the regulatory, so the regulatory can subsequently review, do the rolling review when the data is available. Once all the criteria has been thick, then the vaccine will receive emergency use authorizations, and we needed to follow up all the studies until the follow-up finish. So during the pandemic situation, all the phase of the clinical trials still be conducted, and the vaccine still need to meet the requirement for the safety, immunogenicity, and efficacy. However, we do in parallel, so the development can take quickly than a normal process. So if we retrospectively think about what happened in 2020 about during the pandemic and the development of COVID-19, we can see that the virus emerged in the beginning of 2020, and after the genomic data of the virus available, we start already design the vaccines, and 3 weeks later, the preclinical trials begin, and most of the candidates start clinical trial in May, and phase two and three begin during the August, and rolling submissions start in the last quarter of 2020, and some country by the end of 2020, at least 3 of COVID-19 vaccines has received authorizations. So obviously that's amount of work cannot be done by only single organizations. We need to work collaboratively with a lot of aspects, such as from regulatory, industry, and also research centers like university. So about the Oxford vaccine AstraZeneca itself, also we managed to develop the COVID-19 vaccines at least less than in 1 year. However, the research behind the vaccine ILTA has been done back to 2010. So probably as you heard before, that Oxford AstraZeneca vaccines using a novel platform called viral vectors, which is we use another form of the virus we call which is adenovirus that we got from the chimpanzee to deliver the vaccines for COVID-19. So we use this type of virus as a vehicle to deliver the vaccine component from SARS-CoV-2. So the study about how to genetically engineer this factor has been done in 2010 until 2012, and subsequently we test this vehicle for various diseases that potentially can cause the pandemic, such as influenza, Rift Valley, MERS, chikungunya, Ebola, or Lassa virus. And in 2017, the group in Oxford has managed to create a vaccines against MERS, and we collaborate with NIH in the US to test these vaccines against MERS in rhesus macaque, and to establish that the vaccine is safe, and whether the vaccine can protect macaque, non-human primate, from the MERS virus after the challenge or not. Just after the SARS-CoV-2 emerged, the group published the publication that the vaccine for MERS works. Then when the SARS-CoV-2 emerged, we used the same method to design the vaccine for COVID-19, and also collaborate with the same partner to establish that the vaccines can protect macaque or non-human primate from pneumonia caused by SARS-CoV-2 virus. So, although the development of vaccines of COVID-19 vaccine can be done in less than 1 year. However, the research behind that has been done in many years beforehand. Then we move forward to the clinical development of the AstraZeneca vaccines. Initially, we test the vaccine in small number of populations, less than 2,000 people across 4 sites in the UK. We first test the vaccines in a healthy adult. Uh, this is the publication of when the AstraZeneca vaccine had firstly being administration to human adult. Uh, but also during the clinical trial, the process is not smooth as we want. There is always a challenge during the development. For examples like a hoax from anti-vax that told the first volunteer that received AstraZeneca vaccine has passed death because of the implication of the vaccines. But turn out it's just a hoax. So we need also the participate from the community to counter the hawk news about the vaccines in order to accelerate the process of development of COVID-19 vaccine as well. Once we already established that the vaccine is safe in human adult, we then move to the phase 2 trials that happen across the UK, involving more than 10,000 people. Obviously, we need a resource to do this trial. We use the national health system in the UK network to conduct these trials. We also collaborate with the universities across the UK to deliver the vaccines, and also to monitoring and follow up the volunteers, so we can know that the vaccine is safe and efficacious and also immunogenic. Once we have been established the data across the UK, we also expand the trials across the world. We test the vaccine in South Africa, in Peru, Brazil, and in the US itself, to study if the vaccine also can work differently across the community, across the world, with different genetic background, with different race, with different nutritional status, or other health conditions. We need to test if the vaccines can also work in diverse populations. Obviously, this is a need a collaboration and network across the globe, not only but around a single country, but also from multiple countries and organizations. Gladly, after the trials, we can show that our vaccine is work. Our vaccines can induce human to produce antibody against SARS-CoV-2, and the level of antibody is similar to the people get infected, and the antibody that induced by the vaccine is functional and can neutralize the virus itself. And after 1 year work, we managed to get the data that the vaccines can prevent people to get the severe disease of COVID-19, as shown in this graph, where the people who get placebo, which is meningitis vaccines, has more incidence of getting a severe disease because of COVID-19 compared to the volunteers that receive COVID-19 vaccines. So based on this data, we confident that we can protect people from COVID-19 vaccines. However, the work not stopped in here. We need to think as well about how we can produce the vaccines in a massive amount and distribute globally. So in parallel, a team led by Karina Jo, also Indonesian, working in Oxford, she developed the method to scaling up the production of AstraZeneca vaccines. Initially, we only able to produce the vaccines only for the clinical trial dose, which is only limited to at least 10,000 doses. However, in order to distribute the vaccines globally, we need to produce the vaccine at least million to billion doses of vaccine in across the globe. So we need to scaling up the process production from bench scale to manufacturing scales. And in general, the process take up around 4 steps. The first is upstreams, and to cultivate the cell to produce the virus, and downstream is to purify. Then we formulate the vaccines with some additive like buffer or adjuvant if needed, but in AstraZeneca vaccine case, we don't need an adjuvant. And the last one is fill and finish and put the vaccine into the vials. So beforehand, before the pandemic begins, Karina and her group has been trying to increase the yield of the virus productions using different vaccine target. For example, ChAdOx1 targeting MERS, ChAdOx1 targeting rabies. Based on this data, she able to find the right formulations, the right condition, the right temperatures to increase the yield of the adenovirus of the vaccines. Then when the pandemic happened, she already got that formulation and she needs to scaling up. So she worked closely with the industry that supply the reagents, the consumable part of the bioreactor, for example, to scaling up the process productions. And she managed to scaling up from 200 liters to 2,000 liters in order to produce the COVID-19 vaccines. But also the working not stop by scaling up. We need to think about how the vaccines can be produced in a short time. So we also working closely with other CDMO or contract manufacturing agents across the globe, and also working closely with other pharmaceutical industry to be able to produce the COVID-19 AstraZeneca vaccines with the same parameter, with the same quality across the world. She collaborate with a lot of vaccine manufacturing to do transfer technology, training them how to produce the vaccines, and finally in the first year of the pandemic, we managed to produce 2 billion doses of vaccines in just 1 year. Also, the vaccine itself will not work if the vaccine never been used for vaccinations. In order to get to people to get vaccinated, the vaccine needs to be distributed equally across the globe, because the virus doesn't recognize the national barriers. So there is a role from NGO to help the countries get an access to COVID-19 vaccines. So through the COVAX facility that led by Gavi, WHO, UNICEF, and CEPI, this facility guarantee that the country can get access equally between developing countries with developed countries to get access to the COVID-19 vaccines by kind of work as a mediator for the countries or participants to get an access to COVID-19 vaccines. How the COVAX facility works: first they pull the demand from the participants, and they're consolidating the buying power from every countries or organizations, and use that money to incentivize manufacturers to produce more vaccines, and also to building infrastructures so the vaccine can be diversified. So the vaccine manufacturer can be diversified regionally, because we don't want the vaccines become monopolized by only one country. So we try to diversify. The COVAX facility try to diversify the vaccine manufacturing across the globe. So through this facility, the facility able to distribute COVID-19 vaccine for more than 5 billion doses in the last 2 years. And we also already experienced that Indonesia has a lot of received COVID-19 vaccines from COVAX facility in the first phase of the pandemic. So this is the achievement that we got in the first year of the vaccine rollout, that AstraZeneca has saved most life in the first year of its rollout, because we managed to produce 2 billion doses in the first year, and thanks to COVAX facility, the vaccine can be distributed globally. So collectively, to summarize, all the history, that the vaccine development obviously need a collaboration between different sectors of university, architecture, the lead of the vaccine research, but also we need to collaborate with industry. Industry will be able to produce the
Terima kasih sponsor. Ya.
we need to collaborate with government, public service, and NGO, and also community to participate in the trials. So, in conclusion, that collaboration is very important in the vaccine research, and hopefully, based on the history, we can extract some relevant informations in order to initiate the collaboration in our work. Thank you so much.
Brilliant presentation. Well, ladies and gentlemen, Mr. Indra has conveyed some information, including about the rapid development of COVID-19 vaccine and then the process in developing vaccine. And there is a need to collaborate in vaccine research involving government, public service, university or research center, industry, NGO, public service, and the community. So let's come to the last session. It's Q&A or discussion sections. If you want to ask the questions, you can raise your hand, and you can also chat or drop your question